Hello friends, I am Prashant Katiyar. I am the faculty of the Life Science Academy, the premier institute of GATE and CSIR-NET and ICR-NET exams preparations. So today we discuss about Pentose Phosphate Pathway, and previously we discussed about Pentose Phosphate oxidative phase. Now in today's video we discuss about non-oxidative phase. So previously we discussed how the glucose 6-phosphate, which is most preferable sugar by the cell, which is when this sugar is available in ample amount, directly glucose 6-phosphate consume this sugar and convert it into 5-carbon sugar, that is pentose, that is D-ribose 5-phosphate. So we draw here this scheme. Glucose 6-phosphate is actually converted into pentose, that is D form of ribose 5-phosphate. What is happening in this scheme? This actually scheme is lie under the phase, that is oxidative phase, as we discussed in previous video. Now when this sugar is completely converted into D-ribose 5-phosphate, if further sugar is needed by this cell, then this ribose sugar is automatically following the non-oxidative phase and converted back into the glucose 6-phosphate. Okay, this is non-oxidative phase. Or you can say that is reductive phase. Okay, there is between this compound sugar D-ribose 5-phosphate and glucose 6-phosphate, there is number of other derivatives there between them. So today we discuss about these derivatives. What are the derivatives are involved before forming this glucose 6-phosphate? So first of all, ribose 5-phosphate is actually a 5-carbon sugar. This ribose 5-phosphate combines with xylose 5-phosphate, which is also 5-carbon sugar. But these two sugars are interconvertible to each other in the presence of epimerization reaction, and the enzyme involved is epimerase. When these two sugar, 5-carbon sugar and 5-carbon sugar, both are combined, they form pseudo-heptulose, which is 7-carbon sugar, 7-carbon. And another one is glyceraldehyde 3-phosphate. This one is pseudo-heptulose and this one is glyceraldehyde 3-phosphate. This reaction occurs in the presence of trans-ketolase. Now when pseudo-heptulose is converted into fructose 6-phosphate, and fructose 6-phosphate is converted into glucose 6-phosphate. This is the direct process. When we go through indirect process, when glyceraldehyde 3 phosphate and pseudohaptalose before this sugar is another sugar is formed in this process, that is called erythrose 4 phosphate. This is 4 carbon sugar and this one is 6 carbon sugar. Okay. This reaction occurs in the presence of trans aldolose. Now this 4 carbon sugar combines with xylose 5 phosphate again and form a glyceraldehyde 3 phosphate. Which is called Pentose phosphate also. Now, if we compare this pathway to the glycolysis, what is the similarity between these two pathway? In the glycolysis pathway, when the triose phosphate it forms in the cleavage step, and when this cleavage step moves backward to the glucose 6 phosphate, it follows several reactions. Okay, this is triose phosphate again moving back towards glucose 6 phosphate and form a fructose 1,6 bisphosphatase. Bisphosphate. Okay. Now fructose 6 phosphate 1,6 bisphosphate again converted into fructose 6 phosphate, and fructose 6 phosphate is ultimately converted into a product glucose 6 phosphate. And this reaction, when the erythrose 4 phosphate combines with xylose 5 phosphate and form a triose phosphate, this reaction occur again in trans ketolase. Okay, so this is the whole whole reaction which occur during non-oxidative phase or reductive phase. But one thing I want to tell you, this is the most important thing about non-oxidative phase. What is the need of this phase? Why this phase is occurring? So reductive phase, whenever the reductive phase run, then it is synthesizing something sugar metabolites. So this is the one of the major factor, and another factor is when the cell is synthesizing that kind of glucose 6 phosphate metabolite, it needs some protection from environmental stress like oxidative damage, which is usually occurred by the presence of hydroxyl free radical. This is the ultimate redox oxygen species which damage the cellular membrane and leaks out the cell content leak out the several content and discharge their components into the environment. So this is the main hydroxyl free radical species which causes oxidative damage. During this phase, its utility is it counter the oxidative environmental stress and prevent their cellular membrane from these oxidative damages. Okay, so this is the overall non-oxidative or reductive phase, and during this phase indirectly glucose 6-phosphate sugar is formed, which is utilized by the cell to produce ATP. Okay, whenever the cell when cell consume all the glucose 6-phosphate, now it comes back into the oxidative phase and perform glucose 6-phosphate to form ultimate sugar D-ribose 5-phosphate. As we discussed in previous slide, this ribose 5-phosphate is the precursor of PRPP, that is phosphoribosyl pyrophosphate. This precursor generates nucleotides, nucleic acid and protein and coenzymes, which is the molecular component of the cell. This is all about your pentose phosphate pathway. Thank you, friends. We will, in next slide, we will discuss about how oxygen hydroxyl free radical species affects the cellular membrane and causes this kind of oxidative damage. Okay, thank you.